This will be a brief introduction to the molecular lab. There are many things you can do here. I'll show you a basic sequence of actions that you would take to analyze some DNA. So first you're going to want to choose your type of DNA. So you can filter by name. I was just going to choose tiger. And then you'll click the load pipette button to get the DNA in your pipette. Then you'll want to add your Taq polymerase and tagged or untagged nucleotides. Then you need to choose the correct primers. So there's a big long list of primers, or you can add your own custom primers. For this specific activity, I've added my own primers. So I'll add those two. Then you're going to want to make sure that you added everything correctly. So we have a new feature where you can just hover over one of the tubes and it will give a list of what's in it, which is incredibly useful. And then you'll take your tube, put it in the PCR machine, and start. You can adjust any of the cycles on the PCR if you want to, or you can just run it as is with the default cycles. Then you'll want to advance time because this takes a really long time. So you'll just take it until it gets to complete. Then you'll take out your tube. You can at any point, if you want to, label your tube if you're doing multiple sequences here. So I've run it through the PCR. Now I'm going to show you how to use the gel machine and also how to use the sequencer. So if you grab your micropipette, load it with the DNA, load it into one of the wells in your gel, then you're going to hook up the electrodes. There are standard DNA ladders already set up in here. And so now we will just turn it on. There we go, turn it on. There we go. Okay, and then as we advance time, you'll see the bands start to move. The position of the bands will help you to identify when it has been fully separated according to the mass. Then if we turn on the UV light, we can see all the different bands. You can use our horizontal tool to get the number of base pairs. So that's how you'll use the gel, and you can also pipette out of there later if you want to be able to do further amplification. Now to run the sequencer, you'll load the DNA that you've already prepped into the sequencer, and then you're just going to click Start. This takes a while also, so you'll just advance time until your number of pair sequence stops increasing. So we just run through all of those. Then once it toggles back to start, you know it's done. You can label your tube if you want to. And then you can go through many analysis tools. If you've got multiple sequences here, you can find the differences between them. You can save them to your lab book. All of this can be done within this Zoom window. And those are the basic functions in molecular biology. At any point, if you want to, you can save the lab to create a URL that then you can get out of your My Saved Labs link. So this URL, you can give it whatever custom title you want, and then you'll be able to use this link for anything. Or you can start with any of our lab preset links. These will start the lab at a specific point that then you can use either in our worksheets or just to be able to have good starting points. And those are the basic functions. Hope that helps.